We've been told that pandemics are biological accidents that take years to emerge. But what if that's wrong? New evidence suggests something far more unsettling. The next global killer is already out there waiting, and it's pre-programmed to hijack your cells right now. What if there was never a missing link to the COVID origin story? Not because of a conspiracy, but because the virus may never have needed to evolve to infect us in the first place. It was born with the master key. And if that's true, we're not waiting for the next pandemic to evolve, we're waiting for it to arrive. From the very first day of existence, humanity has had to navigate a biological minefield. We knew that there were viruses out there invisible to the naked eye. We knew they had the potential to wreak havoc inside our bodies and even bring our lives to an early end. We thought we had a certain level of safety. For years, generations even, public health policies, vaccine strategies, and pandemic response systems were all based on a single and important assumption: that there is a buffer between us and the viruses that could potentially wipe us all out. We believed that microbes essentially followed a pre-programmed step-by-step route. First, those viruses would spread through the animal population before eventually jumping into another species like a pangolin or a pig or maybe even a civet. And that's where things began to change. Inside these new hosts, the virus would start changing. Each infection created new copies. Each new copy was another chance for a mistake, another mutation. Most went nowhere, but over time those mutations could stack up, and eventually the virus might stumble onto the exact tools it needs to infect humans. That's when we'd need to worry. Once they gained the ability to infect humans, they could spread from person to person. As they moved through the population, they could trigger powerful immune responses that make us sick. At best, that means pain, fever, and discomfort. At worst, it can cost lives. Scientists called this buffer the species barrier. It was the idea that animal viruses couldn't simply jump to humans. Before making the leap, they were thought to need months, years, or even decades of evolution inside animal populations. It was like a key that still needed to be filed down before it could fit the unique lock of the human body. And that mattered. It suggested that there was distance between us and the world's most dangerous viruses. The species barrier wasn't just a scientific concept. It became a kind of safety net. A belief that we'd have a window to spot a virus spreading through animal populations, to track how it was changing and stop it before it ever reached us. But what if that window never existed? What if the virus was already loaded and ready to fire? Recent research reveals that the evolutionary barrier model might not be anywhere near as concrete as we thought. Because if viruses need to mutate to infect us, then how did the deadliest outbreaks in history skip this step entirely? To understand the implications of this biosecurity blind spot, we have to go back to one of the worst pandemics the Earth has ever seen, the Spanish flu. In just 2 years, it infected roughly a third of the world's population and killed tens of millions of people. It was a uniquely deadly strain of H1N1 influenza, and it unleashed chaos on a scale the world had never seen. In the decades that followed, scientists worked to understand exactly how it happened. They traced its path and developed a model for how pandemics spread. For nearly a century, we thought that model was correct. The pandemic was portrayed as a slow-motion disaster, a chain of events that unfolded over years, not months. If there had been a warning window, the tragedy could have been avoided. But that narrative was shattered around the turn of the millennium. Scientists exhumed and analyzed the bodies of Spanish flu victims from an Inuit community in Brevig Mission, Alaska. The bodies had been perfectly preserved in the permafrost, allowing the researchers to extract samples of the viral RNA. They were then able to effectively reconstruct the virus using modern genomic sequencing techniques. And that's when they uncovered the truth, a truth that rewrote the rulebook on how viruses operate. It turned out that the 1918 H1N1 virus hadn't spent years mutating in pigs before making the leap into human populations. It was believed to have started as an avian virus originating in birds, and somewhere along the way it developed something important: a human-ready hemagglutinin or HA protein that was already capable of binding to human cells. In other words, the virus already had the key that it needed to unlock the doors of the human body. At the very moment of spillover as it passed from wild bird populations into humans, it didn't need to undergo any sort of rapid mutation. It didn't need to multiply to develop the tools it needed to launch a global pandemic. It was a plug-and-play biological weapon found in nature, and that completely defied everything the scientific community had believed about this virus. It called into question the idea of a long warning window and the so-called evolutionary barrier itself. But every virus is different. They mutate and move in different ways. With that in mind, scientists were left with one crucial question: Was 1918 just a bizarre historical fluke, a once-in-a-lifetime example of a virus breaking convention, or is nature mass-producing these plug-and-play weapons as we speak? In 2021, a team of researchers discovered something that shattered everything we believed about how viruses operate. It was at the height of the COVID-19 pandemic caused by the SARS-CoV-2 strain of the coronavirus. There were a lot of different theories about how that particular virus began and crossed from animals into humans. Several studies pointed toward bat populations, so the researchers set out to see if they could find that missing link. They explored the limestone caves of Laos in Southeast Asia, collecting samples from various bat species and taking them back to the lab for analysis. They uncovered a group of wild bat coronaviruses, 3 in particular named Banal 52, Banal 103, and Banal 236 that stood out due to their distinctive genomic similarities to SARS-CoV-2. Further analysis showed that all 3 of them had a specific spike protein that was actually able to attach itself to the ACE2 receptors of human cells. ACE2 receptors act like entry points on the surface of our cells. Think of them as the locks, and the spike proteins are the key viruses used to open them and gain entry into the body. All these viruses possess the necessary key, but that's not all. Using advanced techniques like binding measurements and computer simulations, researchers found something unexpected. The 3 bat viruses behaved almost identically to the original Wuhan strain of SARS-CoV-2. In some cases, they were even better at attaching to human cells, giving the body almost no chance of preventing an infection. One of them, Banal 236, showed clear evidence of being able to multiply inside human cells. These viruses, found far from urban centers, were in some ways better equipped to infect humans than the strain that was at the same time shutting down society. And then it got worse. Unlike SARS-CoV-2, none of the wild viruses possessed a furin cleavage site, or FCS. This is a unique molecular structure that allows viruses to easily fuse with human cells. At the time, COVID-19 skeptics argued that the presence of an FCS was proof that the SARS-CoV-2 must have been modified in a laboratory. It is such a rare feature in nature. It was the smoking gun. But the wild viruses proved that Mother Nature was already producing pre-programmed viral strains that could, in theory, bypass the species barrier. Humans could be infected far more easily than expected, all without the slow, step-by-step evolutionary process scientists had long assumed was necessary. And if nature is building these human-optimized keys in the middle of the jungle, what happens when they find the lock? Middle East Respiratory Syndrome-related coronavirus, otherwise known as MERS-CoV, is one of the most lethal pathogens on the planet. The virus causes a condition called MERS and can produce a range of symptoms from fevers and coughs to respiratory difficulties and diarrhea. The first case was identified in 2012 in Saudi Arabia. Several thousand more have occurred since then, with an approximately 35% fatality rate. There have been several notable outbreaks, including one in South Korea in 2015, though most of the cases have been isolated to the Arabian Peninsula. Studies have found that humans are typically infected with MERS-CoV via contact with camels. If we follow conventional epidemiological models, then the virus found in those camels should be notably different from the one found in the cells of dying human patients. It should show clear evidence of countless mutations that enabled it to pass from one species to another, the very changes that allowed it to survive in the human body. This would confirm the intermediate host theory. That's not what the data shows. When researchers used genomic sequencing techniques to map out the MERS-CoV viruses found in both humans and camels, they discovered a disturbing fact. The two are almost exactly the same. To be more precise, they are 99.9% identical. They're not distant relatives, but biological mirror images. That means that this virus doesn't need any sort of learning or adaptation period to bridge the gap from one species to another. There isn't some vast chasm separating the two, but a tiny, almost imperceptible 0.1% gap. The virus can simply cross the species barrier using the exact same architecture it relies on to infect livestock and other animal populations. It is a fully functional, completely compatible, and capable invader, a microscopic weapon of mass destruction that is more adaptable than we ever thought possible. The implications of this are terrifying from a biosecurity standpoint. This discovery suggests that virologists might have been working on flawed information for years. They've been looking for the wrong warning signs, following unreliable models, and allowing society to sleepwalk its way toward another biological crisis. If animal-based viruses like MERS-CoV are already identical to the human-killing versions, then how many more of them are out there, just waiting to kick off the next pandemic? By the early 2020s, we knew that there were viruses out there that could move from animals to humans far faster than anticipated. Scientists had proved this was the case with the likes of the Spanish flu and MERS-CoV, but they still were seen by many as outliers or anomalies. Then a groundbreaking 2024 study arrived. It showed that these alleged anomalies might be a part of a larger and deeply worrying trend. The study was carried out by researchers at the Bloom Lab at the Fred Hutch Cancer Center, led by evolutionary biologist Jesse Bloom. It was designed to put the evolutionary barrier hypothesis to the ultimate test. They wanted to know whether viruses actually need time to adapt when they jump into humans, or whether they might already be capable of doing it from the start. To do this, the team didn't just focus on a single outbreak or viral strain. Instead, they analyzed large genomic datasets to understand how they behave at a molecular level. They used a technique called deep mutational scanning to study specific viral proteins in detail. Their analysis covered a wide range of viruses, including influenza, SARS-CoV-2, HIV, and Zika. This allowed them to map out how thousands of potential mutations might affect a virus's ability to evade antibodies and infect cells. First, the researchers had to create a so-called pseudovirus. These were safe versions of real pathogens that were able to enter cells but unable to replicate. This eliminated the multiple risks associated with working with live, dangerous viral strains. They were then able to genetically engineer the viral proteins to make their own mutations. It was similar to the ways a viral cell might change or adapt to increase its infectivity and ability to survive in different hosts and environments. The variants were then tested, and researchers measured how well they resisted antibodies and how tightly they attached themselves to the host cells. The data pointed in one direction. Some of the deadliest pandemics in human history had zero adaptation time during their initial outbreaks. In other words, when these strains first entered the human population, they didn't immediately need to evolve to become more effective at infecting us or evading the immune system. They didn't need to, they were already capable of doing that. This exposes a truly systemic flaw in global health surveillance and security. Scientist communities and virology initiatives like the USAID PREDICT program have believed that we should be looking for viruses in the wild that are beginning to evolve human-like traits, ones that could pave the way toward widespread infections. Meanwhile, the more generalist viruses that are able to infect multiple species have historically been viewed as a lower risk. That's because they don't appear to be optimized for human transmission. This research has turned those beliefs upside down. It suggests that by the time a virus begins to evolve human traits, it's already too late. The pandemic has already started. The studies seem to suggest that there could be a number of viruses out there in the wild today, just waiting for human hosts to come along. The one factor that seemed to be working in humanity's favor was that these viral strains were confined to places far from human populations. They appeared to exist primarily in jungles, caves, and other remote ecosystems. If that was true, then it explained why global pandemics have seemingly been a once-in-a-century event. People simply weren't coming into contact with these viruses. The evolutionary barrier model may have been shattered, but a new geographic barrier seems to have emerged. The actual physical distance between human populations and infected animals helps us to stay relatively safe. That was the theory. Researchers made yet another frightening discovery, which sent scientific communities back to the drawing board to reassess their understanding of viral biology. They found that people are actually encountering these kinds of viruses all the time. A study was carried out involving almost 700 subjects from rural communities in Myanmar. They were screened for signs of sarbecoviruses, specific sorts of coronaviruses that mostly infect bats and have the potential to cross over into humans. The study found that 12.1% of the people possessed antibodies for those viruses. That meant that they had been exposed to them at some point in the past. People were significantly more likely to test positive if they worked in industries that involved spending time outdoors or handling raw materials. This included activities like logging, hunting, and harvesting forest products. This was an important finding. On average, over 1 in 10 people in these parts of the world have been infected by wild viruses. Most of it appears to be linked with bats, with some evidence also pointing to pangolins. But when you check out official public health records from those same areas, you won't find reports of major outbreaks. There's no unexpected spikes in atypical infections, no sudden lockdowns or quarantines. But the clues are all there. They're hidden in the blood of these individuals. Those clues are the remnants of these silent test fires of weapons that haven't yet been fully triggered. The old explanations of how viruses work have been well and truly obliterated by this stage. Humans aren't as special as we were led to believe, not in the eyes of viruses in any case. We're just the next step in the chain. Viruses already have everything they need to infect us, and they are actively doing so, albeit in places that don't dominate the headlines. If everything we've seen so far is accurate, then humanity's days must be numbered. Evidence suggests that mankind has been lucky to survive this far with so many potentially deadly viruses circulating in nature. Fortunately, there's one thing that has been working in our favor this entire time: our last line of defense. A biological safeguard that has helped keep humanity alive for generations, one that has allowed us to survive pandemics that killed tens of millions. It's called the interferon response, and it acts like the body's built-in firewall. When a human-ready virus enters the body, the data suggests it has no trouble binding to receptors and entering cells. It already carries the key it needs to unlock those biological locks. That's the easy part, because the moment it enters the body, something else begins: an invisible countdown. If the virus doesn't move fast enough, it won't have the freedom it needs to replicate and spread. Instead, it'll be killed in a matter of hours. And this is where the interferon response comes in. Interferons are a group of signaling proteins also known as cytokines. They're produced by the body in response to the presence of foreign microbes like viruses and bacteria. As their name suggests, interferons are designed to interfere with these invaders. They slow them down, disrupt their replication, and make it harder for them to take hold in the body. They do this in various ways, including actively degrading viral RNA and alerting other cells to defend themselves against the coming threat. Studies show that the majority of wild viruses are entirely neutralized by this initial cellular counterattack. Even though the viruses have the key to pass through the front door, they soon discovered that the house is equipped with a state-of-the-art security system. Because for viruses, the real challenge isn't just getting inside the body or even entering cells. It's surviving long enough to replicate, to multiply fast enough to gain control before the immune system shuts them down. This is the only thing that's saving us from a perpetual state of pandemic emergency. The warning window depends on the virus itself. Some viruses mutate quickly, allowing them to weaken or evade interferon responses. Others are stopped earlier, and it also depends on the body's own ability to detect the invasion fast enough, to sound the alarm and activate its defenses before the virus can spread. That's why those 1 in 10 people in the rural villages of Myanmar have those antibodies. Yes, their systems were invaded by wild viruses from bats and other creatures, but it didn't trigger a massive outbreak or a society-halting pandemic. Their bodies were able to respond in time, and their interferons did the job they were designed to do. The enemy was effectively eliminated just inside the castle gates. But why is nature building these human-ready keys in the first place if we aren't the primary target? It's clear humanity has been following a fundamental misunderstanding of viral evolution for generations. We have spent decades believing that viruses have to adapt to us because we are so different and distinct compared to other animals. Humanity has represented the ultimate prize for these microbes, the pinnacle of their evolutionary pathways. The data tells a very different story. It shows that the most successful viral strains in history, like the 1918 Spanish flu and the SARS-CoV-2 (COVID-19), didn't show any major signs of recent human adaptation. These were what virologists referred to as generalist viruses. They didn't evolve in the wilderness or mutate in dense jungles. They weren't pre-programmed to infect and kill humans specifically. They didn't even require years of random mutations to reach their human-ready form. They simply evolved over time to survive in high-density, multi-species environments. They learned to thrive in places where bats, birds, rodents, primates, and other creatures were present. That gives them an enormous advantage. The viruses spread more easily when they aren't limited to a single species. These viruses effectively come with their very own skeleton keys that allow them to go almost anywhere they please. In other words, people aren't the targets, they're not even the end goals or ultimate objectives, they're just collateral damage. This is what makes the next pandemic an invisible inevitability. The hardware is already there, and it is fully compatible with the human operating system. When nature is creating its own biological weapons, humanity isn't sitting on the sidelines. And when things go wrong, it has consequences. Discover the horrifying truth in viruses that were actually lab leaks. Or watch this video.